We're at the 21st CROI in Boston, Massachusetts, cold Boston, Massachusetts, in a cold room.
It's a very cold
So
room.
we'll stay awake. But we're here, and we need to do that because this is with Michael Malim from King's College in London, who gave the talk on alpha interferon, and it's quite complex. But I want to say to the audience that we're going to do our best to get it to a level where they can understand it for the late term, because it is important work. I think a lot of people have said that to you at the conclusion of your talk, and what we're learning about the way the virus can be blocked. So lead on from there.
So you'd like me to summarize the talk?
Yeah, I think yes. I think that would be the best way.
So the interferons are a family of proteins that animals make in response to infection. And the talk really focused on type 1 interferon, which is a long-established molecule controlling lots of viruses, but the recognition of its effect on HIV-1 is rather more recent. And really the talk tried to highlight, first of all, during acute infection, interferon probably plays a very important role in dampening down virus replication and allowing some host control. But in the long run, as the infection becomes persistent and chronic, in the absence of therapy, it's probably very important in helping drive the pathogenesis of disease by harming the immune system in general.
Mm-hmm. And you showed a lot of correlations between the proteins that are acting to do the blocking.
So the way interferon works is that it induces, when it recognizes a cell carrying the receptor, it induces a whole cascade of proteins, most of which we know very little about how they function. So most of the work now is more about correlations of the whole cascade of genes and interferon itself. So, for example, in experimentally infected macaques with SIV, you see an inverse correlation between interferon alpha levels and viral load during acute infection, but in chronic infection it reverses, and in fact you see a positive correlation between interferon and viral load. And I think one of the big challenges for the field now is to try and understand at a more molecular level what are all the proteins and genes that are involved in these different effects in the cell biology and the effects on the immune response. And actually we know very, very little, which is...
And well, you studied, I think you said there was 11, the first 11 that you're doing?
I mean, we've studied a lot more.
That was just showing it.
I mean,
there
are 900 interferon-stimulated genes in humans. And I think we know a lot about a very, very small number of them, and most of them we know almost nothing about.
So how will this inform the science, and how will we use this science to move the whole field forward?
So it's a bit curious, really. The recognition of the importance of type 1 interferon in HIV infection has actually been recognized rather late. And I think that's probably several reasons for that. First of all, until a few years ago, the large focus of lab-based research was on using immortalized cell lines, which actually show a very poor interferon effect. So it's not until you work in primary cells, plus there's all this lovely work done in experimentally infected non-human primates, as well as human studies that are now sort of all aggregating now into some consensus views about the importance of it. So there's a huge amount to be learned, and I think there's increasing recognition now of these roles in pro-inflammatory effects, deleterious effects to the host. And using animal models, I think I said at the end of the talk, not in an HIV setting, but in a different chronic virus infection model in mice, interfering with the interferon signaling in that system actually gives a very good therapeutic output for those mice anyway. So I think that is a real... So there's the goal just to always understand more biology, because that's always helpful. But I think there is real... appetite really for dealing with these problems of the chronic inflammation, and because of course those are some of the long-term deleterious effects of being infected with this virus.
And all of the work that the envelope, or I should say the whole viral process, going into the cell, going out, and in the DNA transcription. All of this seems to, you seem to have a different way of looking at it, the way the blockings are happening and how there's a relationship between the parts of the way the virus replicates itself through the process of budding and even coming in and being transcribed. It seems as though this is all, is this a new way of thinking, or?
Well, I would say the new aspect, so I think as we've discussed before, the restriction factors have been recognized for a few years. Mm-hmm. But now there's recognition there's a whole another category of genes that probably are not regulated by the virus itself. They are just turned on by interferon. Mm-hmm. And I think that recognition is very recent. I mean, the identification of the one particular gene that I touched upon, MX2, is extremely recent. But as I tried to highlight, there are definitely others out there, because you cannot explain the whole biology of interferon based on MX2. So there's plenty to understand, and the genes we've worked on, that's just the tip of the iceberg. As I say, there's many more.
So you would say this is just kind of like opening a whole new door?
I think so, yes. Yeah. I think so.
Well, I know that it's hard to, and so I guess the key word would be MX2 for now, but to search for, because this is going to be the first step. And then there'll be, like you say, a whole cascade of other genes that may be out there that we are not aware of.
Absolutely, yeah. And I think the key is the dichotomy between the effects on acute infection, where it's beneficial, and the effects in chronic infection, where it really seems to be deleterious. And most likely there will be different genes and pathways that are involved in the two sides of the coin, really. So
and you've shown the correlation between the alpha interferon and the effect and the blocking, and it's all kind of coming together, but huge amount of work, like you say. It's exciting, because it's every time we answer a question, we have 20 more questions. Is that fair to say? Maybe it's 200 more questions.
Yeah, that's one of the beauties of science and biology in general, and it's always very exciting to work on something that's in the context of human health.
Yeah. And this may be translational to other diseases.
Quite possibly. I mean, there's a higher interferon in many inflammatory diseases. I mean, psoriasis is an excellent example.
Mm-hmm. Well, I appreciate you taking the time. This has been exciting, and I'm hopeful that people can learn more from you over time, and from others who are working in this complicated but yet interesting field.
Okay.
All right. Thank you very much.
It's been a pleasure. Yeah. Thank you.